Welcome to Hope in a Dose, the podcast where we explore the science and strategy behind cell and gene therapy. I'm Louise Kearney, and today we're talking about one of the most critical steps in bringing these transformative treatments to patients, early phase trials. Why is this so important? These first-in-human studies are where hope begins. They're the bridge between groundbreaking science and real-world impact for patients who often have no other options. Unlike traditional phase 1 trials, these studies rarely involve healthy volunteers. They involve children and adults living with rare, sometimes life-threatening conditions. This means that the stakes are high and the challenges are unique. Complex logistics, global site networks, specialized dosing procedures, and long-term follow-up that can span decades. Joining me today to unpack these complexities is Fred DeRosier, Vice President, Rare Disease, Advanced Therapies, and Pediatrics at Fortrea. Fred has deep experience in early phase cell and gene therapy trials, and today we'll explore what makes these studies different, why they matter, and how sites and sponsors can prepare to succeed. Hi, Fred. Thanks for joining me today. Today we're going to talk about early phase cell and gene therapy studies. How do you see these different from others?
Well, Louise, I think it's pretty fair to say that early phase cell and gene therapy studies come with a number of unique challenges because of the nature of these treatments. And certainly, unlike traditional first-in-human trials, we're almost always working with patients who have an illness, often quite serious, and not healthy volunteers. And consider, we wouldn't want to edit normal functioning genes in a healthy person. And as these therapies often create permanent, irreversible changes, we really can't expose a healthy volunteer to these therapeutics, for one thing.
And I suppose if we're not looking at healthy volunteers for our first studies, what kind of populations are we looking at?
Well, if you looked at these therapeutics are really very well suited to target genetically driven diseases, these trials then often involve patients having rare or ultra rare diseases. Certainly, this then adds complexity around age, disease severity, recruitment, and some 80% of rare diseases experience an onset in the pediatric range, and for more than 50% of rare disease, these children don't survive into adulthood. So you'll only be working with a pediatric population. And I think, needless to say, the younger your study population is, the more operational complexities you're going to be introducing into the program. And to state an obvious issue with rare and especially ultra rare populations, finding enough eligible patients can be quite difficult. And oftentimes this puts us into very widely dispersed geographies to conduct the study around the world. And as many of these patients are often significantly disabled by their diseases, in addition to being young, any sorts of travel that may be necessary because of this geographical dispersion becomes a problem and an issue for the clinical trial.
And I suppose if we're looking at different patient populations for our first studies, we probably want to think about our study design differently as well.
Yeah, study design is quite different for cell and gene therapies. Unlike traditional first-in-human small molecule studies, cell and gene therapy trials rarely will include placebo or control groups. We still use dose escalation and cohort management as part of the trial structure, but most cell and gene therapies are single dose. So the typical SAD, single ascending dose, MAD, multiple ascending dose design doesn't apply. And the very high cost to produce these medicines, combined with rare target populations and the typically irreversible changes that I mentioned earlier that these therapies affect, means that these studies recruit very small numbers of patients. And while safety and tolerability remain the primary focus of a first-in-human cell and gene therapy trial, we also really need to assess efficacy early on. This means more endpoints, more assessments, and we're aiming to gather as much data as possible from a very small patient population in the clinical trial. Consequently, first-in-human cell and gene therapy studies are almost exclusively phase 1-2, combining traditional phases of clinical drug development. And as I alluded to earlier, we often find ourselves working with multiple sites in multiple countries that may only contribute a single patient. So coordinating the dosing through these cohorts can be a challenge in addition to just simply the nature of the design itself.
Yeah, and with the traditional phase one, you may be looking at just one or two locations. So it sounds like we're looking at more locations and centres to do these studies. So there must be some logistical things that we have to work through. And is dosing a challenge here as well?
Well, I think I can pretty fairly say both are considerable challenges in cell and gene therapy. These therapies require specialized infrastructure for shipping, handling, preparation, and administration. And this whole process for these therapeutics occurs under very strict regulatory oversights, and requirements for this may vary by country to country and even by institution. So that adds additional layers of complexity. Autologous cell therapies, such as CAR T, for example, are the prime example of a complex, tightly managed logistics and product preparation situation within a clinical trial. And the administration process of the therapeutic itself can often be extraordinarily complex when you're talking about cell and gene therapies. A good example would be the MRI-guided intracranial administration of cell and gene therapies for Parkinson's disease, which then requires very specialized equipment, not readily available, a neurosurgical suite, specialist neurosurgeons, neurologists, radiologists, and so forth. So the dosing and logistics that often is found within these first-in-human studies can be extraordinarily complex.
And finally, of course, because of the permanent change, you have to consider that you'll have to follow up all patients in phase one studies for cell and gene therapy much longer than you typically would.
So, yeah, Louise, there's really, another additional and unique, really, challenge to cell and gene therapy studies. The promise of these therapeutics, of course, is that they are going to be single dose and that they're going to be a permanent change. They are going to be really a quasi cure, a functional cure for these patients. And so while this is the promise, this promise of curing previously untreatable diseases with a single dose, we still need to prove and document durability. And given the nature of where we're making these changes at the genetic level in the patient, ruling out delayed adverse effects, effects that can potentially occur many years down the road, simply is going to require us to take the time to observe and follow these patients. So long-term follow-up is not optional. It's a regulatory requirement, and it's a requirement from first patient in the first-in-human dosed all the way to the last patient dosed in a registrational study designed to get market approval. And so this long-term follow-up, really at the shortest end, is typically at least 5 years. And on the longer side of it, it's 15 years. And we've even seen some cases where a recommendation from the regulators is lifetime follow-up of the patients treated. So clearly, following a treated patient from a clinical trial for decades is an inherently difficult challenge and something quite unique to the cell and gene therapy world. And is another place where these therapies differentiate or distinguish themselves from traditional small molecule clinical development programs.
Thank you, Fred, for sharing your insights. Here's what stands out. Early phase cell and gene therapy trials are not traditional phase one studies. They involve rare disease populations, often pediatric, and require global coordination. Study design is different. Single-dose therapies combined phase 1 and 2 structures and more endpoints to capture maximum data from very small patient groups. Logistics and dosing are complex, requiring specialized infrastructure and highly trained teams. And because these therapies create permanent genetic changes, long-term follow-up can be 5 years to 15 years, and perhaps even a lifetime is mandatory. Thank you for listening to Open a Dose. Subscribe for more insights on advancing cell and gene therapy, and keep an eye out for further episodes of the Hope and Dose podcast. I'm Louise Kearney. See you next time.